Controlling gene expression, the Arabinose Operon. So prokaryotes use these segments called operons, and it's essentially a group of related genes that are controlled by a single promoter. So you've got these genes that need to be transcribed, and they've got this particular promoter. The genes would be related, and therefore they'd want to be transcribed together, and it can end up as one long strand of mRNA. So bacteria normally prefer to, and again, bit of a generalization here, but bacteria normally prefer to catabolize glucose for energy, and so the pathways involved in doing that, the glycolysis pathway with all of the associated enzymes in there, are the constitutive genes that are always going to be on, normally going to be on anyways, because they're normally able to get some glucose to do that. So like hexokinase is the enzyme that has to be made and made from the genes expressed for hexokinase. However, sometimes glucose is low, and some bacteria have the ability to metabolize other sugars, and so they have other pathways to metabolize those other sugars that are coded for by other genes. So for example, they can break down lactose or arabinose as other sugars, but they need other pathways to do that. And so as long as glucose is present, no need to worry about the proteins to break down lactose and arabinose, especially if they aren't present to begin with. You don't need these proteins, these enzymes, to break down food you don't have. And so they tend to be turned off. However, if there is some arabinose and maybe there isn't any glucose, then the bacteria would want to be able to use its genes to break down arabinose that are normally off. It'd want to be able to turn them on to build those machines. So the Arabinose Operon, araBAD, is referred to as it's referred to. These structural genes involved in the processing of arabinose, being able to break down arabinose, they make up a portion of this operon. But as you can see, it is a single portion of this operon here. So we have this big segment here that does include the genes that we're going to be talking about, but it's also going to control some other control sites associated with that, because we don't want the bacteria doesn't just want to make the enzymes necessary to break down arabinose. Only when it needs to does it want to be able to produce those proteins, and therefore it wants to have this off most of the time. But we're going to look at when it is able to turn on the transcription and then translation and production of the proteins for the consumption of arabinose. So the Arabinose operon is composed of the promoter regions and structural genes. The structural genes ABAD, so these guys here, these are the ones that make the enzymes to break down arabinose. They normally want to be off, but in some cases the bacteria want to be able to turn them on. Activator sites, we have this CCAP and this paraBAD. These are two activator sites that we're going to be looking at. These are the genes over here, as we mentioned. They code for proteins that break down arabinose. This is the promoter sequence, and so it's called pBAD, and this is where RNA polymerase would bind if we are to transcribe the araBAD genes to break down arabinose. These guys here are regulatory sites, and so things can interact with these sites to stop or to help RNA polymerase bind to pBAD so that we can make these araBAD genes that would break down the arabinose. This arac here is actually another gene further upstream that codes for a protein that can stop or help RNA polymerase bind to pBAD. So you can sort of see how these are all connected to each other, and how they get expressed and interact depending on the conditions is going to dictate whether we have whether the bacteria have the actual machines to break down arabinose or not, the enzymes from these genes. So first thing is that the arac protein, the one that's upstream from there, can act as a repressor. So this arac protein that is encoded for can act as a repressor, and it stops RNA. So here's the araBAD genes that would make the machines to break down arabinose. RNA polymerase, if it were to read it, the bacteria would be able to make those enzymes to break down arabinose, which is good if you have arabinose. Normally, the bacteria is using glucose though. So this arac protein stops the transcription of the arabinose genes. So no araBAD transcribed, you have no arabinose metabolism because the machines to break it down are not present, and that's normally the conditions that a bacteria would want to be in. So these arac proteins, the little blue guys here, they essentially sort of form this kink in the DNA, and they stop RNA polymerase from binding, and therefore the arabinose breakdown enzymes never get made because the genes do not get transcribed. Arabinose sugar acts as an inducer for the arac protein. So arabinose is the metabolite that could potentially be an energy source. Normally it's not there. Normally the bacteria is going to be using glucose. But if it was there, it actually acts on the arac proteins. So it's an inducer for that protein, and it actually turns that arac protein into an activator. So as an activator, activators do what activators do. They work on those control areas upstream of the gene. And so if arabinose is present, you want to express this gene. You want to make the machines that can metabolize the arabinose. So the arabinose itself acts as an inducer, and it turns the arac former repressor into an activator. And as an activator, it's going to be able to turn on that gene. One more part though. Glucose itself, if a bacteria has glucose and it has arabinose, it might as well just stick to breaking down the glucose. So glucose, when it's present, is great to be using, but when it gets low, there's actually this production of CAMP, and it's going to build up in the cell. CAMP is another inducer for another activator protein, this CAP protein. And so imagine this CAMP, when the glucose is running low, it gets produced. It combined with this other protein to form this other activator. So we get this second activator that can activate or work with the operon, to get RNA polymerase to bind and to get the transcription of those PBAD segments. When arabinose is present, making the arac protein, former repressor, into an activator, and glucose is low, making another activator, the CAMP activator. This is when we get the full transcription of the araBAD genes. So let's look at what that looks like. So this would normally be a repressor. When arabinose is present, it gets turned into an activator, lands on the control site, and can help the RNA polymerase bind. But also this CAMP is going to land on this other site, and now you've got this double promoter region. This is going to allow RNA polymerase to go through and make the mRNA necessary, the mRNA coded from those genes, the mRNA is necessary to make the proteins to break down arabinose. So you can see when the cell would actually start producing the proteins needed to metabolize arabinose. To summarize it, if the cell has high glucose and high arabinose, there's going to be very little, so it's somewhat repressed because you don't have this other activator. You only have one activator here, and so you don't have a lot of production of these arabinose genes, and therefore, because there's glucose present, the cell doesn't really need to break down the arabinose. However, if there is, wrong one, let's do this one first. Low arabinose, you definitely don't want any production at all, and that's where we actually have this, imagine the picture we looked at earlier, where this arac protein is still acting as a repressor, so you have it acting as a repressor, and if you have high glucose, you do not have this activator here, so it would be gone, just like in this first case here, you don't have it, and so now you don't have an activator and you have a repressor. It is going to be super repressed. There's going to be no machinery produced to break down arabinose. Again, if you have low arabinose and you have low glucose, you still have the arac acting as a repressor, so it's still going to be low or to no production of arabinose, because again, if you don't have arabinose, there's no point in making the machines to break it down. However, if you do have arabinose, you do get this promoter and you do get this promoter, and therefore RNA polymerase is able to bind to the promoter site, and we are able to get that production of the genes necessary to break down arabinose.